They make us happy. They give us energy. They help us to grow as people, as a business, as a country. We are top sector horticulture and starting materials. Our strength is to innovate, to make sustainable. We co-create together with companies, knowledge institutions and governments. Every day, we are committed to green habitats, health, trade, economy, employment, energy, and of course, flowers. Top sector horticulture and starting materials, together towards a sustainable future.
Hello and welcome here at this live online innovation meetup about root zone breeding. We have an exciting few hours ahead of us where we will inspire you with fresh knowledge from the world of science and where we will try to give you some actual face time, some networking opportunities with people from both industry and academia, so together we can shape the future of horticulture. I'm glad I don't have to host this alone here today. I'm joined by Clemens Stolk. Clemens, good afternoon.
Good afternoon.
You are officially titled an innovation broker. You are fascinated by improving the impact of science and technology, specifically in horticulture and agriculture.
Yes, for sure.
And you designed this program here today for us. Why is it important that we spend the next few hours thinking and talking about the root zone of crops?
Yes, well, the roots, they are very important because that is where plants acquire their nutrients, their food, their water resources. And without success below ground, you cannot have success above ground. But roots are difficult to study, so usually they don't receive that much attention. But if you want to be successful also as a breeder, I think it is vital that you also look at what happens below ground.
Exactly. So it's half of our crop. Nobody looks at it. So it's important today we zoom in and we dive underground. What is your personal fascination with this topic?
Yeah, well, I'm also, apart from the function you mentioned, I'm also a program manager of the program Better Plants for New Demand, which is a public-private research program consisting of about 100 research projects, all in the area of plant breeding, co-financed partly by TKI and also by companies participating in it. And I think of those 100 projects, I think just only one looked at roots.
Okay.
And I thought, well, if they are so important, maybe spend some time on that for the discussion. Yeah.
Well, thank you, and thank you for being my co-host throughout the program. You will help me and our viewers guide through the building blocks, because we have some exciting building blocks for you. Soon we will hear an official welcome from the director of the top sector horticulture and starting materials. After that, we will listen to Professor Malcolm Bennett, who will show us his latest insight about how you can phenotype the root zone. And we'll talk a little bit about that in a second, what that means. Then next up, obviously, at least here in the Netherlands, it's about lunchtime. We are aware we have a very international audience. We'll dive into that soon. And during lunch, we have our first experiment where we try to give you some actual face-to-face networking time. We'll hop out of this live stream into a separate video conferencing with 10 people, moderated by our moderators who are ready for you to guide that conversation. Then after that, around 2 o'clock, Amsterdam time again, we come back here for a live stream and listen to our second scientist, Dr. Erik Visser, who will tell us about his work in understanding the roots of our crops very well. And then later, all the way at the end of the afternoon, we will try to imitate or actually have a real networking event like we would have if we would have a physical innovation meetup here today. So stick with us, be curious, and dare to experiment, because we believe that we're actually going to have some true fun and also create some meaningful connections this afternoon. It means that it's important that you participate with us, and we have a few means to do that. When you look at the time allocated to our speakers, over 25% of that time is devoted to your questions, to answering your questions. And you can send in your questions to us here in the little studio in the Hortiversum via Slido. That's a tool that you see on the website here, embedded on the right side. You can send in questions yourself, or you can promote questions from others with a thumbs up if you really want to prioritize that question, if you also want that question to be answered. And we can ask questions to you. And currently we're doing that. Maybe if you've clicked around, you already found it. I've placed a small survey there with 3 questions. And if you haven't done so, make sure you scroll all the way to the bottom and hit the send button, because then the results come in. And so far, let's see on the screen, we see that 52 people have already submitted. First question is, in which sector do you work? We're curious. We're trying to bring together science and industry. And let's see how we succeeded, Clemens, this morning. We see of the people who voted, about 37% from science, 52%, over half from industry, and 15% denotes themselves as other. We're very curious to learn what that is. And again, if you're just joining us now, go to the right side of your screen, answer the 3 little questions, and make sure you hit the send button to submit the question to us. So we've proven the fact that we brought together a diverse crowd. Right, Clemens?
Yes, for sure.
Exciting. The next question is, where are they from? I already announced we have an international crowd today. Let's see. Obviously, most of us are from the Netherlands. In this little word cloud, the Netherlands, and even the Netherlands is very large. But, and this is where it gets interesting, we have people today with us from Belgium, Italy, the Philippines. Good evening to all of you there. Saudi Arabia, Spain, Israel, Germany, France, Ireland, and Italy. So literally an international crowd all over Europe and into the Far East who are curious to learn about what's new in the root zones when we're breeding the crops of the future. So welcome to everyone. Stay tuned and let's have some fun together. Last question. Obviously, we live in very special times at the moment, both in our personal lives, but also in our professional lives. So we would love to know, what do you currently miss most in these Corona times? And can you summarize that in one word? And that also is now on the screen as a word cloud. And the largest thing, Clemens, is here, colleagues. We miss our colleagues.
Yes.
What do you miss most in these Corona times?
Yeah, also the interaction with other people. Yeah. So because that is where you will receive inspiration, where you get new ideas. Also, actually, the idea for this event also came from some conversations I had with people. Yeah. And it is all now much less natural.
Yeah, it is less natural, but we're going to try to get as close as we can today. Other words we see is the meetings. Today we're live online with this innovation meetup. Interaction, coffee. Well, if you miss coffee, the beauty of being online is that you can run to your kitchen anytime you want, get a fresh cup of coffee, unless you have really poor coffee in your house, of course. But, well, maybe it's time to invest in that. Friends, people, yeah, contact. In fact, that's what I think, well, at least if I talk for myself, we miss the most as well. But again, this afternoon, we're going to try to bring as much of that as we can. Inspiring content and face-to-face networking. Enough said, this is how it works. If you haven't done so, please submit your questions through the talks when they happen. And with no further ado, I'd like to introduce the operational director of the top sector horticulture and starting materials. He's here with us in the Hortiversum, ready to welcome you all. And Michiel Roelse, take it away.
Thank you, Gerrit. Well, a very warm welcome from me as well. Mabuhay to the people tuning in from the Philippines. As top sector horticulture and starting materials, it is indeed a pleasure and a privilege to be hosting this very special event today. As explained earlier in the movie, the top sector is a triple helix organization, initiating and executing a knowledge and innovation agenda in triple helix connection, so with the government, research institutions and companies. And in addition to setting the agenda, we also have an annual budget to fund innovation of 35 million euros, both public and private money. And over the last few years, we have accumulated a very strong portfolio of innovative projects. Our main instrument to distribute our money is the annual call for proposal. I will discuss the call and maybe give you a few tips later in the program when we come back. And any information you might need on the top sector, the activities, the research programming, can be found at topsectortu.nl. And any information on our extensive valorization program can be found at Impact 2025. But if I may, I'd like to say a few words about the corona crisis, because this is unlike anything we've ever experienced before. And I've heard people around me describing this crisis as a total reset, and I wholeheartedly agree. I'm not trying to downplay the fact that this is first and foremost a human crisis, a human tragedy, where hundreds of thousands of people have lost their lives. My wife and I have lost an uncle to COVID-19, and he was only in his 50s. tens of millions of people have lost their jobs and their chances of prosperity or even security. So this is a human crisis. But someone once told me that the problem is not in being knocked down. The problem is in not trying to get up. So that's what we're going to have to do. We were here before the crisis. We've been knocked down to here. It's up to us to make our way back to the top. And the first and foremost, what we need, the single most important thing to climb back to where we were, is innovation and R&D. Invest in innovation and R&D. That is the main thing. That will help us answer this crisis. And since we're climbing anyway, let's not stop at where we were. Let's climb a little bit further. Let's go to a place where we can have more sustainable business models, a more sustainable way of working, a more sustainable way of life. And as we are gathered here this afternoon, we have people from companies, from research institutions, policymakers, government officials. We can do that. We can be our own innovation frontline, if you will. And together we can make this happen. That is what the triple helix is all about. And that is what the top sector is all about. Thank you.
Wow. Thank you very much, Michiel Roelse, Director of the Top Sector Horticulture and Starting Materials. We are the innovation frontline. And with that, I think we have an assignment here this afternoon to be curious, to reflect on everything we see and hear about what it can mean for our ideas, to share these ideas as soon as they come up in the networking opportunities, and see if indeed we can bounce back even better than we ever did. Well, thank you for stretching us there, Michiel. So we come to our first keynote speaker of today, Clemens, Professor Malcolm Bennett, who's a professor in plant sciences from the University of Nottingham. Mm-hmm. When you were designing the program for this afternoon, why did you find him a key player who should be on our agenda?
Yes, well, Malcolm devoted, I think, his entire career into researching exactly what we will be discussing this afternoon, plant roots. Mm-hmm. The first part of his career, I think he looked mostly at molecular pathways involved in root development, and did groundbreaking research and published in high impact journals with those findings. And especially, I think, the last decade, he has been involved in developing and designing methodologies, technologies, even coming from, say, medical background, to really study, literally look at plant roots as they grow in their natural habitat. So that is one reason, I think, why we invited him.
Nice. And when I read his bio, I saw he's also in the top 1% most cited animal and plant biologists. So yes, he's a real celebrity in the field. We're very honored to have him with us. Last year, he published a review paper with the same title of his talk today, Uncovering the Hidden Half of Plants Using New Advances in Root Phenotyping. Now, before we will listen to Malcolm Bennett, maybe it's clear to understand phenotyping. It's a word I don't come across every day in my kitchen. What are we talking about?
It has to do with observing, so that you observe what the organism is expressing, what it is doing. It can be measuring yield. It can be measuring certain compounds that are that occur in the plant. So you measure what is on the outside. So your phenotype for eyes would be brown eyes.
Exactly. So it's something you can observe,
yes,
as opposed to gene typing or genotyping, where you look in the inside.
Yes, exactly.
Very clear. Yeah. Very interesting. Now, for those watching, please understand that Professor Malcolm Bennett has divided his presentation into 3 parts. He will first talk about field phenotyping challenges. So what are the main challenges in the field? Then he looks for roots grown in a controlled environment. And thirdly, we will look at some exciting new innovations in this phenotyping field. And after each part of his presentation, we're going to take some first questions from the audience. So if anywhere during the talk you are listening and you hear something of interest, make sure you make use of the fact that you can type in your questions on the right side of the screen, submit them to us, or if your eye catches a question that you find interesting, hit a thumbs up, so we will ask that question in the little 5-minute transitions. With that, I saw him already quickly on the screen here. I think he's with us. I would like to give the floor to Professor Malcolm Bennett in Nottingham.
Hi, good day, everybody. It's a great pleasure to join you. I'd like to talk today about advances in root phenotyping. I'd like to point out that this is really not just a personal presentation, but I've drawn on a lot of expertise from colleagues, collaborators from around the world to try and give you the most rounded assessment of the current state of the art. When we reflect on the future challenges facing agriculture, we obviously heard a little earlier about the impact of COVID on the sector. But we should also reflect on other, even longer term impacts, the impacts of diseases, nutrient, water scarcity, climate change, all these challenges which are, in this graph illustrates, may impact negatively, in this case, wheat yield. Obviously, the activities of agronomists and breeders are going to be instrumental in driving up the yields to deliver the projected demands that are required in this case for wheat production. But this is true also in many other sectors, particularly horticulture. And so clearly this is very, very important. One of the overlooked areas in terms of plant breeding, as was discussed earlier, is the hidden half of plants. And if we can imagine, for the last 10,000 years, we focused on the upper half of the plant ever since the Fertile Crescent experiments and other areas around the world have led to crop improvement. But the hidden half of plants has really only been started to be addressed in recent decades. You could imagine that there are many traits which are very important for improving crop function. For example, as we attain greater sustainability in agriculture, we want to apply fewer fertilizers. And so we want to optimize the root system, for example, to improve nutrient capture. And I'll talk a little bit about this in one of the examples I'll give in the first section. Root architectural traits really play an important role in terms of nutrient capture. For example, if you can see my cursor, you could see root surface area has a huge impact on capture of nutrients like phosphate, which accumulates in the topsoil. So does root angle. So these traits really matter in terms of capturing nutrients in the natural agricultural setting. And our team over the last 25 years has looked at the genes that control these processes. So for example, we spent a lot of time looking at the genes that control root angle, also root hairs, and more recently, we've done a lot of work looking at the genes that control root branching. So this is looking from a very molecular perspective. But as we appreciate, over the last 10 years, I've really started to embrace studying roots in the natural environment, in soil. And this is where phenotyping is really important. And I'm going to divide today into 3 sections to reflect where and when we study the root traits. So I'm going to talk about some of the examples that we're doing with colleagues around the world on field phenotyping. So this is actually going out into the field in the correct setting and studying traits in the field. I'm also going to talk in the second part of my presentation about how we can control the environment in the laboratory or greenhouse and how we can study traits in that context. And finally, I'd like to share with you what I think are some of the most exciting new innovations in root phenotyping and arguably plant breeding. And to stimulate some discussion. Okay, so let's talk about the first part, which is field phenotyping. And I'm going to talk to you about 2 projects. The first is focused in Southeast Asia, and this involves colleagues at IRI in the Philippines and also colleagues in Thailand. And we're looking at the importance of developing rice varieties which are less drought sensitive. Drought is a major problem and major threat to global rice production. This is particularly the case within Thailand and the major rice production area of the Northeast Plains. We started a project, this is led by Jonathan Lynch, a colleague who's based at Nottingham and the US. And it was to address one of the problems with rice, current varieties of rice. Here we have a schematic of rice, and the majority of paddy rice, shallow rooting, and they're obviously prone to drought stress. We are obviously interested in developing deeper rooting varieties with a variety of traits such as steeper root growth, reduced numbers of nodal roots, for example, reduced lateral roots. And also, in addition to these architectural changes, we're also interested in looking at the effect of anatomical changes. So most of you are familiar with the idea of root architecture, but we should, if we delve down deeper into the actual anatomy of the plant, that can also have a big impact on plant performance. So the work was performed in Southeast Asia at various sites across Thailand and also at Iri. And the variety of rice germplasm was exposed to a number of drought conditions. And we used, just to illustrate some of the methods that we applied, we used a variety of ways to phenotype in the field. One of the simplest ways is so-called shovelomics. This is a technique where you simply dig up the crown of the plant, as is shown in the picture below. And you can get a lot of information from this in terms of root angle, a number, etc., which can really be very, very informative for breeding programs, as I'll illustrate later. You can also perform other techniques such as soil coring, where that allows you to determine the branching density of roots deeper in the soil profile. You can also use various processes such as baskets, which allow you to remove the plant intact from the soil and assess, for example, root angle. Just to give some examples of the work that applying these techniques and the insights it provided. So we looked at a variety of shallow rooting rice varieties and also deeper rooting rice varieties. And interestingly, when we grow these varieties in well water conditions, there's no performance impact of either. But if we apply a drought stress, there's a clear improvement in performance for the deeper rooting varieties based on shoot dry weight. And this can result in quite significant changes in biomass. Obviously, the deeper rooting varieties are able to access water more effectively. Another surprising phenotype was at the anatomical scale. And as I mentioned before, anatomy can really impact crop performance. So if you look, for example, at varieties with small xylem vessels, so these are water transporting vessels in the root, which move the water from the soil through the root to the shoot. So varieties with small xylem vessels had no performance impact, but when you actually apply drought stress, you saw a clear improvement in performance of the ones containing the smaller xylem vessels. And the reasoning behind this is that you're removing less water from the reservoir of water. You're being more efficient economical with your water. And this, in a way, provides a greater use, eking out of the resources. And this will obviously lead to improvements in yield as well and yield stability. So this is just one example. This is the first project, and this involves colleagues in Penn State, Erie, and several Thai universities who are pictured here. I'd like to tell you about another project which is very much focused. It's called DEEPer. It's funded by the ARPA-E program at the Department of Energy. And they have an amazing program called Roots. And the idea behind this is that if they were able to grow maize, for example, which if it was able to root 0.5 meter deeper, you could bury 2/3 of the CO2 emissions from the US transportation. So there was great interest in ways to do carbon sequestration in this way. And so the idea behind this $30 million program was to develop ways to develop crops with deeper roots. And so we were one of the large programs. This is led out of Penn State, and we're the UK partner. And the idea behind this is that we want to look at the maize varieties and identify deeper rooting varieties based on their architectural and anatomical traits. It's known, for example, that some anatomical traits have a big impact on terms of how deep plants are able to root. For example, here we see an example of two varieties of maize. One, the lower panel, has air spaces, and these are so-called parenchyma. The net effect of these air spaces are the roots are using less energy to maintain the cells and therefore able to invest that carbon in additional deeper roots. So the targets of this program involved a number of different approaches to a phenotype, and I'd like to focus on one, which is to excavate and image crown roots. As you could imagine, digging up roots is a back-breaking, challenging process, and one of the programs within our project was to develop the so-called root robot system. And this is allowing us to automate the root phenotyping process. So rather than doing this all manually, there is a team of engineers developing a root robot. And this allows the maize stalks to be removed from the soil. The soil is washed off, and this is in this mechanism. The crowns are then placed within an imaging system, as is shown here. And we use a series of cameras to image the root system. And we use a variety of approaches to image the root crown. As you can see here, the root crown is oriented upwards, and then multiple image angles allow you to develop a three-dimensional image. So on the left panel, you see the real root. On the right panel, you can see the computer reconstruction. This is beautiful work done by colleagues at the University of Georgia. You can see that the root is actually cut in half. And by doing this, we're able to uncover the internal structure using this approach. A variety of different image analysis algorithms are used to capture key traits such as stem width, numbers of lateral roots, angles, etc. And what's really exciting about this automated process is that many of these automated features which are captured using the software agree closely with those taken manually. So this provides a real opportunity to use to automate and simplify the phenotyping process. Just to give you one example of how we're using this for agricultural importance, we've looked at a wide variety of a diverse collection of maize. So this is at the WIDIF panel. It's composed of about 800 varieties from the US, the University of Wisconsin. We've used this to look at crown root angle, variation in crown root angle. And we've used this in combination with a technique called genome-wide association studies. And this has allowed us to identify several loci which appear to be important for crown root angle. And one of these genes we've knocked out, we've mutated, and we see a very interesting change in the overall angle of the root systems as shown here. So in the case of the mutant, when we knock out the gene, we can see a far steeper root angle. And interestingly, when we do greenhouse and field studies, you can see that the angle is steeper. And when we actually look at their field performance, this appears to confer an improvement in nitrate uptake. This is really important because we think, and this is based on modeling, simulations of these root systems architectures, we anticipate that steeper rooting allows more time for the nitrate, which is moving with water, to be captured by the root system. So this steeper rooting trait is a really attractive trait when you're dealing with capturing water and also water mobile nutrients such as nitrates. So here we have a really nice example of how we're using phenotyping to identify a novel gene. This gene's being patented now by ourselves, our colleagues in Wisconsin, Penn State, and Nottingham. And as you can see, it delivers clear improvements in field performance. So this work was part of the DEEPer program, and this is led by Jonathan Lynch at Penn State, but it also involves maize breeders at Wisconsin, led by Sean Kepler, some brilliant image analysis colleagues, Alex Bush, and myself and colleagues at Nottingham. I'm happy to take questions as I pause for the next part of the talk.
All right, thank you very much, Professor Malcolm Bennett, live with us from Nottingham. Very interesting presentation, and if you joined us later, this is the time where we take questions from the audience. And so far, Professor, we have one from Joost Kos, and Joost is backed up by 6 people who say, hey, we want to know that as well. But actually his question is more to us. Do we record the presentation? Yes, Joost and the others, we do and we will, and this entire broadcast will be available later on demand as well. So, Clemens, we've seen an impressive root robot. We've seen some exciting 3D imaging. What is your first reflection and perhaps question to Malcolm after this?
Yeah, well, I was of course impressed indeed by what we saw already. When I saw the locus in the I mean, the diagram showing the QTO, I was thinking, well, exactly what is it? But I understand that it's being patented, so we have to wait a little bit until it becomes public. But one question which I had, all examples that you showed so far, Malcolm, were from arable crops. If you think of the Dutch setting, we have many horticultural plant breeding companies. They also are attending this event in large numbers, so they find this an important and interesting topic. Do you have anything to them to say, or do you think there's relevance in these topics as well, also for horticultural crops? Because they have nothing to complain, those plants. They receive water and nutrients on demand, let's say. So what would be your message to them?
I would say that they face the same challenges in terms of developing more sustainable crops. I'm aware that in Holland there is this circular economy model where you're looking at reducing waste, creating more sustainability. And so I would argue a lot of the horticultural crops which you're describing, the same pressures still apply as arable crops. You want to reduce water use, you want to reduce nutrients, you want more efficient capture and less waste. And so these generic insights that we're developing for arable crops could also be applicable within a horticultural setting. Clearly, if you're using a hydroponic system, different rules apply. Yeah. But if you're using a soil setting, then the same it applies.
Nice. Thank you for explaining that and creating even a broader relevance of these technologies. Questions are coming in, Clemens. First one I'd like to take, backed up by 2 other viewers, is from Guusje Bonnema. Guusje is asking, is root phenotyping in soil possible in all soil types? Because clay, as we have a lot here in the Delta in the Netherlands, seems to be a problem. What is your experience there?
That's a very good question. Practically, it can be quite challenging in clay. A lot of the phenotyping we've done is deliberately done in sandy soils. Yeah. Clay can be challenging for techniques such as chevelomics, because obviously you have to remove it without damaging. So this is where the need for non-invasive approaches are really important, and automated approaches. And that's why we're really excited by the root robot. This allows a great deal more automation and therefore hopefully higher throughput. Yeah. But it is a challenge.
Any experience with the root robot in clay soil or not yet?
This is being used in Anger in Pennsylvania this summer. And the soils there are a real mixture. And so we're using it at several different sites and different soil types to really assess this.
Yeah. Well, thank you. I'm going to take one more question before we're going to let you on into the second part of the presentation. It's from Corné Pietersen and already backed up by 7 other viewers who all like to know this as well. What can you tell us about the contribution of the root microbiome in the root development and architecture?
That's a really good question, and this is very much a developing field. Clearly, the root microbiome has a huge impact on the performance of things like legumes. You know, obviously, they form symbiotic partnerships. Many plants, over 80% plants, form mycorrhiza interactions, which are important for nutrient access and efficiency. So the microbiome is extremely important. These are just 2 of the classic examples. There are many other species that we're still just learning about. And there is lots of really interesting work being done in industry using microbial seed coatings to give plants a head start. The science behind that is still not fully understood. But so I think this is a really developing area. And if you think, you know, the plant, we tend to focus on the genetics of the plant and how that impacts, but the physical structure of the soil, as alluded by the last question, and the microbiome, there's almost this trinity of factors which are impacting root performance, and they all play a part.
All right, so big praise to our audience who's immediately diving into the right angle and the right corner with questioning you. For now, we're going to park it because we invited you to do a second part of the presentation where you look at imaging techniques for roots grown in controlled environments. After that, we do another short Q&A. So if you're watching, you have a question or you want to promote another question, keep up the great work. Professor, take away the second part, please.
Okay, thank you very much. So as we move on, we're going to start looking at controlled environments. There's obviously within a horticultural setting, this is probably more relevant. And I'm going to show you a number of different ways to phenotype roots using a controlled environment, which are largely greenhouse-based approaches. So I'm going to show you there are a number of different ways to image root traits for plants. I'm going to show you some very simple ones. These are soil-free imaging using a 2D-based approach. I'm going to show you how we can also use aeroponics for root phenotyping. Soil is very important. Soil has, as we've heard before, has a huge impact on root architecture. And so one simple way of still bringing soil into the phenotyping is to use so-called rhizotrons. These are thin, thin layers of soil which still allow the roots to be imaged. And then there are other ways which are now being developed for high throughput imaging of roots in pots. So I'll briefly talk about that. And then I'll spend the majority of the time talking about work in our team that's using X-ray micro-CT and which is now allowing us to image the roots throughout a crop's life cycle. Okay, so the very simplest way to phenotype roots would be to remove the soil completely and actually look at the way the plants grow in 2 dimensions. And this is possible, and this helps generate quite a lot of insights about early seedling traits. And so this has really been an area that has been pioneered using blue paper. And you can see here how we can grow large numbers of plants. And when they're grown on filter paper, they can grow for about 2 to 3 weeks. And this allows us to capture their root systems architecture, simple features. And we're developing, for example, ways to automate the capture of the images. We're using deep learning algorithms, which I'll talk a little bit later about, software, which allows us to automate the capture of this information and, for example, recognize features such as the main roots versus the branches. And this can give a lot of information in terms for breeding programs. And this helped us identify genes that control these critical root architectural traits. Another way of imaging, and this is developed by our colleagues over in Louvain in Belgium. It's a beautiful system using aeroponics. And this was developed in Xavier Drey's laboratory. And you can see here the roots are able to move around, and you're able to image the roots as they grow and branch and develop. And this could be applied to horticultural crops quite easily. But obviously, this is in the absence of soil. So there's a lot of information you can capture from this. It's a lot easier to do than to actually image in soil. And this, so this is able to image many hundreds of plants in a day. So it's very high throughput. The next step up is to include soil. But in this case, you're still forcing the root to grow in 2 dimensions in a thin layer of soil. This is so-called rhizotron, or rhizoboxes. And this is a really nice setup developed at Germany by the Jülich Center, which is a real pioneer in root phenotyping. And you can see they've developed a beautiful facility here of many different rhizotrons, which move and then are imaged in this central imaging room. And this allows you to capture how the roots grow, the roots and the shoots grow over time. And a lot of information can be delivered. Obviously, the roots grow across against the soil. The rhizotrons are grown at a certain angle, and this allows the roots to be imaged against the surface. Other ways to do this, which are a lot higher throughput, are to use them in combination with automated systems. So there's been a lot of interest in these high throughput imaging systems, originally pioneered by a company called Lemnitech, and there are many others which are employing them. And this allows you to grow thousands of pots of plants using conveyor belts. And these have originally developed to image shoots, but very recently, companies have developed ways to remove the outer layer of the pot and expose the roots as they touch the surface. And this allows a very nice way to image roots and shoots simultaneously. And this work is really being pioneered at Dijon in France as part of the Phenome program, the French national program led by Francois Tardieu. So all these techniques are really nice. They're high throughput. They give you a snapshot of the root system at various stages of plant development, but they're still relatively 2-dimensional. And they don't give an idea of the 3-dimensionality of soil. And this is where our center, the so-called Hounsfield facility, this is named after Godfrey Hounsfield, who was a local man to our university, who got the Nobel Prize for inventing X-ray micro-CT. And we have invested in a number of different micro-CT platforms through local UK and European Research Council funding. So we have 3 different platforms which I'd like to discuss with you today. I'll talk mainly about the mid-CT, which can do large pots, and then I'll finally finish this section describing the large CT. If you imagine you have an object you want to CT scan, for example, a plant in a pot, when you micro-CT image, you capture thousands of 2-dimensional sections of, in this case, the soil. And then you have to stitch it together to generate a 3-dimensional root architecture. Now, obviously, this is challenging because these are huge data sets, which terabytes of information that needs to be interrogated, and the roots have to be segmented away from the soil. And this has really been the major challenge for using CT. And so I'd like to emphasize a point here, which is true for all phenotyping approaches. You have to build an image pipeline. And the image pipeline isn't just about capturing the image. You also have many other steps in this process. So the acquisition is really step 1. And the second step is to recover the root, so separate it from the soil. And then you have to develop algorithms to quantify these traits. All this information needs to be put into a data management system. And then this information has to be modeled and simulated and knowledge generated, insights into this. So capturing the image is really just step 1. And so we've spent a lot of time investing in developing software that allows us to go from the original two-dimensional X-ray image through to automating the creation of these three-dimensional volumes. And this has really gone through a whole evolution in in 2012 using object tracking approaches. And very quickly we realized that deep learning approaches were far more effective. And for example, you can get commercial software which is able to recognize routes, but this is very, very slow and requires a great deal of manual segmentation. Very recently, we've been developing deep learning algorithms and ones which allow us to, through the use of training data sets, to generate output images which are very compelling. So this allows us now to reconstruct images faster than we can acquire them. And this is a game changer because this really does allow us now to scan and reconstruct in real time. And this allows us now to be able to reconstruct the roots, to uncover the roots from the soil in true three-dimensional fashion. And this allows us then to capture lots of really important traits, such as the root count, the tip, the number of branches, the total area which is actually captured. And so these are key traits which could have great agronomic and horticultural importance in terms of optimizing nutrient capture. We're also using this approach as a discovery tool. So some of the work we do really looks at how we know very little about how roots respond in the soil environment. And we're constantly discovering entirely new ways that plants adapt to their local conditions. And this is an example which is called hydropatterning. And if you can imagine you're growing a plant in a pot of soil, if it's in contact with moisture, it will branch quite normally. But on the right-hand side, you can see a root is growing down this macropore. The root branching is completely different. And this gave us a really interesting insight. It allows us to conclude that roots actually branch, but the branching of roots is very much influenced by their hydraulic landscape. So when you grow a plant along the surface of a biopore, it will only branch into the soil. It will not branch into the airspace. Similarly, and this is important for horticulture, when you're culturing the plants on agar, the roots will preferentially grow into the agar. And these are responses, adaptive responses, which we've recently elucidated the molecular basis of. I'm not going to talk about that today. But we've identified critical genes in papers in Science and Nature Plants, which describe, which control these important responses. We've also been upscaling the CT approach to be able to look at plants throughout their life cycle. So we'd like to look at how plants grow from start to finish. And this required us to create something which we call the Hounsfield facility, largely funded through the European Research Council Advanced Grant to myself and the team. And this is a Lego image we use for science fairs, which shows you how the lab looks like. So we have this we've taken the lid off the lab, and you can see the large open space where we have the 3 CT machines. We have an adjoining greenhouse. And the greenhouse then has a large room-sized CT, which is able to take these columns. And this movie shows you how we're able to grow, in this case, wheat, throughout their life cycle, and periodically scan it at different developmental stages. And this has allowed us to actually visualize the root system throughout the life cycle, identify key points in this. So as you can see here, we have the root, which is turned 360 degrees and then rescanned at different points. We take approximately 8,000 sections, optical sections, two-dimensional sections through the column, and then we have to reconstruct this. And as you can imagine, this generates terabytes of information. And this requires, obviously, hugely involved segmentation processes. So we initially started doing this manually, and now we've been applying the deep learning algorithms to be able to do this automatically. This is the readout of one of those scans we did in the case of wheat, where we're looking at how a wheat plant grows throughout its life cycle. Several things, so we've scanned this column multiple times, and this has allowed us to reconstruct how these roots grow. The very first roots you're seeing here are seminal roots that come from the embryo, and they're the pathfinders. They're able to grow very deep and acquire water. Later in the season, as the plant tillers, you can see this flush of roots which originate at the top of the plant. And this really is about capturing as much nitrogen as the grain is filling. So this has really been really insightful for us to understand the dynamics of how roots grow, in this case, within wheat. We've also been working with colleagues in Saudi Arabia to look at, for example, date palm. And this is a nice example that illustrates how we can use we can rescan the same plant multiple times using CT, and we're able to build up a life history and how the root system is able to grow and develop. And you can see, in this case, you can see some remarkable changes as this adjusts to its, in this case, desert environment. And using this approach, we've discovered entirely new organs, and this is some lovely working collaboration with an ex-Wageningen colleague, Ikram Blilu, who's now based at KAUST. And this is a nice example which illustrates the knowledge which can accrue from using X-ray CT. If you're interested in this particular aspect, we've got something called the Hidden Half, which is an information resource which gives you lots of images of CT of different species, many of them horticultural, and they're all freely available. So please have a look at that. So this work was done through a large multidisciplinary team funded through the ERC Award, mathematicians, engineers, computer scientists, as well as plant biologists. So I hope I've convinced you that there are many ways to image roots in controlled environments. I'd like to flag up that all these different techniques I've described today are all platforms within something called the European Plant Phenotyping Network. And this is a network of facilities across Europe in which you could gain access to. 13 of these facilities actually perform root phenotyping. And I've illustrated a few examples around France, Germany, Belgium, but there are many others. These are open. This is an infrastructure award, and this is open to industry as well as academia to apply to. The next call to have access to the platforms I've described is June 2020, and to start the projects later in the year. Okay. So I'll stop there, and I'm happy to take questions.
All right, thank you very much, Professor Malcolm Bennett. Very exciting slides and images and videos you share with us there. Just a few sentences back, I noticed you used the word convince. I hope I have convinced you. And at that moment, I thought it was appropriate to launch a quick poll amongst our audience. So if you're watching, direct your attention to the right side of the screen where I've asked you the following question. To which extent does your company or organization already thoroughly study the root systems of your crops? And it's a 1 to 10 scale. 1 means we never study the root systems of our crops, and 10 is we fully understand and study the root systems of our crops. I'll let that poll go for a little bit. We'll look at the results in a minute. People are voting. Keep doing that. In the meantime, Professor, I'm curious to learn. You talked about the hidden half and all the imaging you have done. What would you say is your most important learning of the root zone that you have obtained from all this 3D imaging and this motion-driven imaging that you can do now?
I think my favorite discovery, well, there are probably 2. One is so working with an American group, we were able to demonstrate the roots are able to respond to their environment particularly their water. The water availability has a profound effect on branching. And this hydropatterning mechanism I described was really the basis for this. And we've done lots of work looking at how roots respond to water. It's fair to say that roots probably respond to water more than nutrients. So water availability is critical. As many of you know, water is, if there's no water, then the nutrients are not soluble, they're not available. So roots have developed a number of really interesting adaptive responses to acquire water. And this is just one of them. And there are many more to be discovered. The second one, which I haven't spoken about today, is we've done a lot of work more recently understanding how roots respond to compaction. And we've recently elucidated the mechanism for compaction. So we now know how plants respond to compaction, the underlying mechanisms, and how we can actually overcome soil compaction. So we're very excited by this discovery, and because it could offer the opportunity. Soil compaction is a huge problem, particularly within field settings, modern agriculture. So this is obviously something which we're really excited about, and hopefully rolling out to breeding programs.
Interesting. Thank you very much for sharing that. That gave our audience the opportunity to actually answer the poll. And I think your choice of words was appropriate. You used the word convince, because here we see a bit of a scattered landscape. About 43% have submitted their vote now. But Clemens, when I look at this, I still see quite a heavy side on the left, right? So rather never or a little study of the root systems than those who actually say 8, 9 or 10.
Highest is almost never, eh? So
almost never. So we have a whole world to win here.
Well, it's at least a good thing to talk about now today.
Exactly, yes. We've proven that. Thank you for submitting your votes. I'm quickly looking at the time, so I will allow for one question at this moment related to the last part. Let me select it. Quite some questions coming in. We need to take time of that. Which one has the most likes at the moment? It's this one, I think. Maria Venus, who was also the first to submit a question. She wrote us, are there cultural management manipulations that will change root architecture to favor a certain root growth pattern? For breeders, it is also interesting to see which materials are responsive to it, if there are any. What is your reflection on that, Professor?
That's a really interesting question, Maria. We know, for example, that the distribution of nutrients, like phosphate, is not equal, at least within natural soils. So phosphates tend to accumulate in the topsoil. So what we've observed in a number of papers, and others have, that developing varieties which are able to have shallow rooting systems to forage in the topsoil is really important. So, for example, beans do this very efficiently. Beans in South American soil, South American soils are notoriously low in phosphate, but by foraging in the right zone, you're able to acquire that nutrient. So having that knowledge about the soil and the way that nutrients are distributed, I think is really important.
Exactly. Clear. Very important. And yes, there are ways to influence that as well. With that, and looking at the clock, I want to save some of the questions to the last Q&A break. So if you're watching and your question hasn't made it to the top of the list, please, all other viewers, review the questions and prioritize. Because with no further ado, Professor, I'd like you to take us to the third part of your presentation, where you have some more exciting future technologies to share with us. So please, go ahead.
Okay, thank you. I'd like to return to one of the points I was discussing in the first section, which is the idea of phenotyping. We're all fixated with phenotyping at the architectural scale. So everyone, I hope, can appreciate now, after 30 minutes of listening to my presentations, the importance of root architecture. So you could imagine that the angle of the root can have a profoundly affect its ability to acquire water, nitrates, phosphate, branching, etc. So architecture is clearly important. I don't need to convince you of that. Probably anatomy is a more novel target because obviously it's a scale which is beyond out the naked eye and requires obviously new technologies and new ways to examine. And I'd like to talk to you a little bit about this because I think this is a really exciting new scale which we should embrace. I've called this anatomics. It's a new form of omics at the anatomical scale. And obviously, if we're going to study anatomical traits, we need to develop ways to image, but also analyze and extract knowledge. And I'd like to talk to you a little bit about the pipeline that we've developed. And this is very much driven, again, through the deeper program that's led out of Penn State by Jonathan Lynch. And I'd like to talk to you initially about how can we phenotype at the anatomical scale. And this is a development by my colleague Jonathan Lynch initially, and this is the very first instrument that was developed at Penn State called laser ablation tomography. And this, I refer to this as LAT. And basically the idea behind this is you have an incredibly high-powered laser which shaves off the surface of a object, in this case a root, and then images. So you have this, you can build up very nice three-dimensional images of root samples brought back from the field and subsequently analyzed. And this allows you then to develop really quite sophisticated three-dimensional breakdowns of the particular organ you may be interested in. The reason I'm obviously going to talk about roots in this context, but I've got some other data which you might be interested in discussing later on, where we've also used this technique to look at, for example, shoot and seed tissues. Now, obviously, you're generating, again, terabytes of image information, and this is even more complicated than, I would argue, the root and soil. You have to identify individual objects, individual tissues within these images. And this is where I think the revolution in image analysis in terms of artificial intelligence and particularly neural networks is really taking off. So this is some lovely work done by Eze, who's a PhD student, my colleague Tony Prigmore in the Computer Science Department of Nottingham. He's using deep learning algorithms to train and then automate analysis. And you can see here are a number of LAT images on the left, and then you've got the annotated images, and then you've got the CNN, the automated network output images. And these are some early examples of that. More recently, this is now, the algorithms have been much improved. And as you can see, we're now developing, we're picking out many different features such as the xylem vessels, the parenchyma, these air spaces I talked about before. So we're able to automate it using these deep learning algorithms. One of the really exciting things, obviously these algorithms are being optimized in this case to analyze maize root images. But there are one question I'd like to ask you is, which species are these sections from? Well, only one of them is actually from maize. All the other ones are actually from related cereal species, sorghum and pearl millet. And the algorithms we've developed in maize to analyze maize images can also be transferred using something called transfer learning to start to the classification of pearl millet tissues. They're not optimized, but they're still able to capture a lot of information. And so we're very excited about this, the opportunities to do transfer learning. The reason it's really promising is you only need a training set of about a tenth of the size that you would need initially if you've already trained a neural net on a particular set of images. One of the things that can really improve the performance of these neural nets is to improve image quality and then apply transfer learning. And recently we've been developing what we call LAT version 2. So we've taken it to the next stage. This is a new facility which has just been built at Nottingham. And so this is a $600,000 instrument, which funded by our research council in the UK. And this allows us, this uses the next level of laser. It's an athermal laser, which generate truly incredible image quality. And we're optimizing the lenses we use on this, but this is allowing us to look at unprecedented levels of resolution, all the way down to, for example, looking at layers, specific layers within the root images. These samples that I show you here, these have been taken from a field trial in Senegal in West Africa and brought back to the lab at Nottingham and analysed. And you can see we're generating incredible detail about root architecture and specific features. So we're using this as part of a program to phenotype roots to accelerate breeding of stress-tolerant crops with novel anatomical traits. And we're using this LAT in combination with the deep learning approaches to identify, again, using genome-wide association studies. So we're extracting, we're analyzing we're imaging, we're using deep learning to extract the information from the images, and then we're using this information in combination with GWAS to identify the underlying genes. And just to show that this works, here's an example of the GWAS analysis in maize, and we're identifying a whole series of different genes in maize, which control, in this case, root cortical rencomer. And these are really promising genes which appear to be associated with processes which you'd imagine would be associated with rencomer formation, such as programmed cell death and reactive oxygen species and hormone signaling. Okay, so what I'd like to finish off is just by reviewing some of the key themes covered in today's seminar. I hope I've convinced you that phenotyping techniques like laser ablation tomography and computed tomography are really helping uncover the hidden half of crop performance. I hope I've illustrated that we can't stay within a single discipline like horticulture or plant science. We have to reach out to and adopt a multidisciplinary approach to really start to look at the hidden half. So active engagement and uptake of approaches with other disciplines is really important. And I think the deep learning illustrates this. This is transforming image analysis, and the horticultural sector really needs to embrace this. Pan-European initiatives such as the European Plant Phenotyping Network and emphasis, and I'll say a little bit about emphasis in a second, are really helping us pool the tools and resources we have within Europe and make sure that Europe is a leader in this field. Emphasis, I should flag, is a European science framework initiative, and it's developing an integrated pan-European infrastructure of instrumented facilities. These are field as well as controlled environments. And what's really exciting about this initiative is that we're developing European-level data information systems. And this is really important. I hope one of the key messages that's come across today is that acquiring the images is just the first step. Analyzing, developing algorithms and managing that data is 90% of the effort. And then obviously developing it and evaluating and disseminating knowledge in novel technologies is also an important part of this work. So I'd like to just give use the last minute to give a plug. If you're interested in some of these root topics, Nottingham is hosting a meeting which happens every 3 years. It's called Routing 2021. We had to delay it from Routing 2020. And so it's now going to happen in May next year. And we're running this simultaneously with the International Society of Root Research meeting that's taking place in Colombia. And we're developing this really interesting way of sharing keynote speakers and poster sessions. So if you're interested in some of the topics I've talked about today, we would really like to welcome you to Nottingham, either in person or online in May next year to also delve deeper. Okay.
Thank you very much, Professor Malcolm Bennett, live with us from Nottingham. And thank you for rooting 2021. We hope we connect again. And talking about connecting, I want to highlight some viewer behavior that I just spotted. Because in the chat box on the side, where people can submit questions, Guusje Hazelmansmeyer has done two fun things. First of all, she added the word potato next to her name, so I don't think that's actually her name, Meyer Potato, but they're significant or highlighting which crop she is dealing with. She's posting a question, and then Joost Slijderink is actually using the reply function in this Q&A box to actually already point out the ReadyMax project of the University in Copenhagen. So this is where the true networking is happening. Yes, we're going to relay some of your questions in the next 5 minutes to the professor. Those questions with the most thumbs up. So this is the time to review the list and let us know which questions you want us to ask. But in the meantime, you can already exchange with each other as well. So, Clemens, shall we look at the most voted question now?
For sure.
It's a question that arrived a little bit back already, Professor. It's from Joost Kos, but it received 12 thumbs up already. So a lot of people are interested to learn this as well. Because he's writing, most pot and bedding plants have optimized fertilization in specific designed substrates. Do you have any experience? It's a very broad question. Do you have any experience to share in this area? Because especially in the beginning of your presentation, you started with showing the phenotyping in the field. What learnings can we transfer to the world of substrates?
No, that's a really interesting question. I guess the horticulture industry is facing questions about developing more sustainable and environmentally friendly materials to which to propagate plugs. And I think clearly these are challenges you face, and I would urge you to really think about developing types of materials which are obviously sustainable. In terms of my knowledge of the horticultural setting, I really have to hold my hands up and say I don't have a huge amount of experience. So I really can't, I would be misleading you if I claimed I did.
Okay, well, we'll keep that in the back of our minds when we take Mark Aarts' question, which is quickly going through the ranks, because again, he also points out that specifically for horticulture, water availability is often not much of an issue, which could mean it would be possible to identify an optimal, quote unquote, root system architecture and set a target for breeding. And then he's asking, is that indeed true? And if so, how could we do so?
That's an interesting question, and I have been involved in a number of projects with companies which are looking at rootstocks. So, for example, tomato rootstocks play a really important role, even in greenhouse-based hydroponic systems. And so, rootstocks are a really large part of that sector. And there's a lot of interest in optimizing root architecture. In terms of increasing surface area, to have more efficient acquisition of nutrients. So I think that this is a really exciting area. Rootstocks is a multi-million pound industry, and obviously developing improved root systems is an important target.
So the short answer is yes, it is possible. We could
Yes.
target a breeding. Yes, yeah, interesting.
It is, and for example, we've done some work with Syngenta and developed rootstocks, which were much improved in terms of their performance, and I think they're going to market.
Interesting. Thank you for sharing. Another question is from Leonard Zorn. Yes, and it's moving the ranks as we speak. More people are giving it a thumbs up. Leonard is writing, you've shown us some examples of shoot part trait quantification. And have you found any interesting correlations between the root and the shoot traits?
That's a really good point. I mean, if you think evolutionary, the root and the shoot, they kind of evolved quite different, separately. So many of the genes, so if you think about, evolution over the last 450 million years, the initial land plants didn't have roots, so they came later. So many of the genes which control root traits are distinct from shoot traits. However, there's obviously harmonization of the size of the shoot to the root through metabolic signals. And this is an area, these sink-source relationships are fascinating. And I think it's an area that we really only know very little about at the moment. So I think it's an exciting area of research. Clearly, you don't want to develop a crop which has a huge root system and a small shoot system. There are many ways in which the plant optimizes the size of its root system to ensure it's large enough to support the shoot system. The mechanisms underlying that really require a great deal more research.
Exactly. So it's an entire field for those 40% scientists joining us today. This is an area where we would like to direct their research efforts, I guess. Yeah, definitely. Well, Clemens, I'm looking at the clock, so I'm handing the last question or the last reflection to you. We've been listening to Professor Malcolm Bennett. What is something that really sticks? What is your key takeaway of this presentation of the professor?
Well, I think there's a whole lot still to learn and to do. I was struck especially by when Malcolm Bennett explained that you need a whole pipeline of, say, data acquisition, data.
We thought it only started with cool
exactly.
imaging technology.
So, exactly. So what I was thinking about, that means that you have to really build a whole interdisciplinary team. So you cannot work as a biologist alone anymore. You need to have a lot of people around you with other disciplines, and you'll need to be able to collaborate with them as well in a team. So I think that requires additional skills. That is
bring in the data scientists into the horticulture and plant breeding fields. Yeah.
Exactly. Yeah.
Interesting. Thank you very much. Yes, Professor?
Yeah, that's a very interesting observation, Clemens. The many large teams now are obviously recognize that. Biologists tend to get fixated on toys, but clearly that's, as I said, is step one. The French, for example, they've spent tens of millions on developing data management systems, and that is being rolled out to the rest of Europe. And I think that's very exciting, the way we can share these investments through initiatives such as Emphasis, because that's really the way to go. Different countries have emphasized different aspects of their investments, but through sharing, I think we can be so much more successful. And that's true not only for countries, but also for disciplines.
Well, thank you. And I think those are wonderful closing words and a nice transition into what's up next here in the program at this innovation meetup. So with that, on behalf of Clemens, the top sector, horticulture and starting materials, and myself, we would really like to thank you for being with us live from Nottingham this afternoon. And we are curious to learn more about your future work. Thank you very much, Professor.
Thank you.
Because now, dear viewers and participants, it's time to come into action. For the next 13 minutes or so, let's make it 12 by the end I finish this instruction, you have a short break to get your lunch ready. At least if you're based in the Dutch time zones, it's lunchtime now. With that lunch, you come back to your computer and it's time to get some face-to-face structured networking done. We have divided you into different groups of a maximum of 10 people. When you received the email with the latest information, you clicked on the first link to get into this live stream. Well, if you're watching this, you succeeded in that. But now revert back to that email because there's a second link that brings you into the Network Tables platform. When you hit the platform, first thing we would like you to do is quickly edit your profile. You see that on the left side of your screen. Upload a picture so people actually can see who you are. Maybe add some additional details because not only today, but also for the next 2 weeks, you can go back to this platform, look at all the profiles of the other delegates who are in this innovation meetup. You can click on their profiles, get in touch with them. So make sure you dress up for this virtual networking party as well. Create your profile first. Then the next step is in the top right side of your screen, you see a button called My Sessions. And you've all been assigned to one network table with a group of 10 people. When you click on that, you enter a Zoom meeting, a Zoom video conferencing meeting, where you find a moderator. And this moderator has prepared 3 statements that he or she would like to discuss with you during the lunch. First statement is, and I'm reading them out for you, so you can either take a photo of your screen when you see them, so you have them with you, or you can start thinking about it while preparing your lunch. First statement is, breeding root traits is not necessary. Basically meaning that everything claimants have compiled today is nonsense. Everything I want to know from the crop can be measured above ground in the harvestable parts. Statement number 1. Statement number 2. We cannot do without high-tech methods and deep learning algorithms if we want to further advance our knowledge of root growth and plant breeding. Something to discuss amongst each other, reflection on the presentation you've just heard. And the third statement, and we've said to our moderators, these are talking points. It doesn't have to be that you touch on all the statements, but potentially you get into the third one, which reads, it's a long one, stay with me. Instead of breeding for root systems that are good at one task, such as capturing nutrients or withstanding drought, we need to breed for highly plastic root systems, which means those that are capable of adapting to local circumstances at that moment. And this will require to design completely new phenotyping strategies. Are you with me? Anyway, the most fun of this is that you actually get to get some FaceTime with 9 other delegates in this meeting. So go over there now. If for any reason you're experiencing some technical troubles, somehow you cannot find the link, you cannot get into the Zoom, you're being called away, I mean, make the best effort you can because your other table companions are waiting for you. But if anything fails, don't worry, because at 5 minutes past 2 o'clock, again, Netherlands time, we will continue here in this live stream in the first link in your email with our second speaker, which will be Dr. Erik Visser, who has a fascination about looking at the individual traits of crops. So with that, I think we're all there. If you run into any troubles, my colleague Sibren is currently in the Slido chat posting his mobile phone number. Sibren, I'm missing a 6. Maybe you want to repost that again, because otherwise we can never call you. I'm going to delete that message quickly. He's reposting his mobile phone number. If you have a problem, please phone him and he'll try to help you as well as possible. So, short break. Make sure you're in the breakout session by 1:30 the latest, which is in 9 minutes now. And we wish you some fruitful, exciting networking ahead and look forward to welcoming you back here in the studio. Right, Clemens?
Yes, for sure.
Thank you for now and see you back in a little bit more than half an hour.
They make us happy. They give us energy. They help us to grow as people, as a business, as a country. They feed many mouths, nearby and far away. They inspire. They provide color and fragrance. They connect and they bring together. Our strength is to innovate, to make sustainable, to work together, to look beyond borders, to invest in solutions for now and the future. We co-create together with companies, knowledge institutions and governments. We have the knowledge to enable growth, the technology to make progress, the drive to offer opportunities. We have the willpower to improve, here in the Netherlands and worldwide. We are convinced that we can do more with less, that we can achieve more together with fewer natural resources. Innovating for here and for the world calls for sustainable solutions and smart technologies, with consideration for our environment, with a passion for people, for our growth and our planet, and for the significance we have with all of our expertise, our training opportunities, our innovations, and our international connections, now and in the future. We are top sector horticulture and starting materials. Every day, we are committed to green habitats, health, trade, economy, employment, energy, and of course, flowers. Top sector horticulture and starting materials, together towards a sustainable future.